Okay, welcome everybody to this joint seminar between the Departments of Chemical, Environmental, and Materials Engineering and the Department of Civil and Architectural Engineering. So today we have the honor of having as our speaker, Ms. Afeefa Abdool-Ghany. Afeefa received her BS and MS degrees from Florida International University, and she's a Ph.D. student studying with Dr. Solo Gabriel. So I'll turn it over to you, Afeefa.
Thank you, Dr. Chin. So today I'll be presenting to you enterococci in the nearshore environment and properties of sargassum during composting, which are two of the projects for my PhD dissertation. The first project, which is really looking at enterococci in the nearshore environment, was funded by the village of Key Biscayne and done in collaboration with NOAA, with some colleagues out at NOAA. With the second part of the project, which looks at the properties of sargassum during composting, we collaborated with the City of Hollandale Beach, and the project was funded by the Hinckley Center for Solid and Hazardous Waste. We also worked with colleagues out at the University of Florida just to complete some extra economic analysis for that part of the project. So an outline of what I will be presenting today, it consists of three major parts. I'll go into the background of what is enterococci, just overarching problem of enterococci in the nearshore environment, and how a beach advisory is issued. The second part, which we'll talk more about the first part of first project in my dissertation, would look at the levels of enterococci at the marine beach, and we were given the unique opportunity of sampling during the COVID-19 shutdown. So we decided to look at before, during, and after the bacteria levels during these time periods to see how this affected the beach environment. And the last project is just looking at evaluating compost as a means to recycle sargassum. So what exactly is enterococci? Enterococci is commonly found in the gut of most mammals and birds. It is an opportunistic pathogen that could cause millions of humans and animal infections annually. So enterococci is used as a fecal indicator bacteria by the EPA to say whether or not a recreational waterway is polluted with feces or that originate from humans or that can originate from animals. And this is only associated with humans and animals, but can be associated with just like natural regrowth in the environment as well. A little bit, it's used because it's readily culturable and it can predict whether or not a waterway is polluted. So how are beach advisories issued? So all of the beaches throughout South Florida are monitored, or even Florida, are monitored by the Florida Healthy Beaches program, and they use enterococci as the indicator bacteria to say whether or not a waterway or recreational waterway is deemed good, moderate, or poor, which are the three categories that are used to classify a waterway. So within these three categories, they look at enterococci colony forming units, or CFUs, per 100 mils of water. So what classifies a waterway as being good, it ranges from 0 to 35 CFU per 100 mil. Moderate is anywhere from 36 to 70 CFU, and poor is anywhere from 71 or greater. And whenever there are two consecutive samplings of 71 or greater CFU, then a beach advisory is issued. So just to give you an idea of what enterococci, cultivable enterococci can look on a plate, you can see down here, each of these small blue colonies is a colony forming unit, and that counts as one positive towards trying to fit into these three different categories. And all of the analysis are based on this cultivable method for this study. So why is enterococci a problem, or why is this bacteria in the environment a problem? So there are 18 beaches that are monitored in Miami-Dade County in the Florida Healthy Beaches program and through the Department of Health in Miami-Dade County. So in a study that was published last year by my colleague Laura Tomanchak, these 18 beaches were separated into regional zones to evaluate whether beach advisories are specific to one beach or is it regional. So in the picture to the far, in the graph to the far right, you can see the heavy black line indicates the beach advisories that are issued by the Miami-Dade Department of Health. From 2012, you can see that there has the number of advisories are based on the measurements of enterococci in the water, and it has increased drastically. In 2016, it is important to note that the threshold that is outlined by the Department of Health changed. So it changed from 104 CFU to about 70 CFU. So we wanted to look at whether or not this change was due to the water quality itself, or was this change due to the threshold levels being changed from 104 to 70 CFU. So to the two graphs on the left, you can see from about 2015 and onwards, there has been an increase overall in the bacteria levels throughout all of the beaches in South Florida. Even though these beaches were broken down into different regions, you can see overall there has been an increase across all of the beaches. So what exactly is causing this increase in bacteria at the beaches we see in South Florida, and what can we do to help minimize the bacteria that's found at the beach? So that brings me to enterococci. So what are the sources of enterococci, known and unknown, in the environment that we can identify? So we know that enterococci can come from dogs when people walk their dogs along the beach. It can come from birds. You know, you can't really get rid of birds from the beach environment. So they can come from birds when they walk along the shore and they pick into the seaweed. It can come from just human shedding when people and individuals bathe in the water. And it can also come from sewage, any sewage leakage, any if a wastewater treatment plant is not operating as it should, and there is sewage spilled into the waterways, enterococci can be introduced into the environment in these ways. So within the beach environment, we have different matrices. So we have water, we also have sediment, and we identified within this study macroalgae as being an important matrix for enterococci. A lot of research has been conducted with water and sediment, and there has been extensive research done with these two matrices, but there's been limited research conducted in macroalgae or seaweed that can make its way into the beach environment. So the study site for the first project of my dissertation is the village of Key Biscayne. So the village of Key Biscayne has been experiencing about 8% exceedance in 2019, and they really wanted to know where these high levels of bacteria are coming from. So close to the village of Key Biscayne, not too far off the shore, you have the wastewater treatment plant. So we did consider looking at the wastewater treatment plant, but within some modeling done with my colleague Peter Salwell, we concluded that the wastewater treatment plant was not impacting the beach as much as, let's say, other sources of enterococci. So if the wastewater treatment plant is operating as it should, which given this modeling that we conducted, it has, so you need a very high level of bacteria from the wastewater treatment plant to actually impact the shores. So we concluded that the wastewater treatment plant is not the main source of bacteria to the beach. So if the wastewater treatment plant is operating as it should, where is this bacteria coming from? So we looked at the shore environment and we tried to figure out and pinpoint down where exactly the bacteria may be coming from. So the village of Key Biscayne, specifically Key Biscayne Beach, is one of the beaches that has been monitored by the Miami-Dade Department of Health since 2000. So there is an extensive data set to look at and to analyze for Key Biscayne specifically. Some of the methods that we have for this project is we collected physical and chemical parameters such as the pH, salinity, and turbidity, because these are important measurements when looking at the enterococci, because these do affect the amount of enterococci that can be found in your sample. We also looked at weather measurements. For enterococci, tidal height and wave height is very big in terms of the amount of enterococci that can be found in the environment. We also looked at the temperature of the different sediment zones that I will talk about in a little bit. And we also looked at any prior rain to the study site. And since we were interested in looking at humans and animals as being a potential source of enterococci, we did take measurements of the amount of people that can be found to the beach, 100 feet to the right and left of the transect that was set up. And with this project, we looked at culturable enterococci within the 3 matrices of water, sediment, and seaweed, but we also looked at microbial source tracking and used different markers to see what can we pinpoint specifically as being the source of enterococci. To give you an idea of the transect that was set up within the Key Biscayne beach, we collected 3 different water samples. So we collected a sample in the ankle depth, knee depth, as well as waist depth. And I want to point out that the waist depth is also where the Miami-Dade Department of Health collects their samples. And if there's an exceedance of the 70 CFU, two exceedance of the 70 CFU or higher, then that's when the beach advisory is issued. So we wanted to collect a sample along the same, where the Miami-Dade collect their sample as well. We also collected 3 different sediment samples. So we have the supertidal zone, which is that nice white sand that you enter as soon as you walk onto the beach, that nice grainy sand. We also collected an intertidal sample, which is where the water and sand and the shore meet. And we also collected a subtidal sample, which is the sediment collected right underneath the knee depth sample. What is really interesting about this beach, and as you can tell from the picture, is the village of Key Biscayne does integrate their sediment and their seaweed. So you can see the drastic difference in what the supertidal zone looks like versus what the seaweed integration zone looks like. You can see that rich, organic, almost organic-like sand when it's mixed with the seaweed. So we collected an integrated sample whenever we saw the shore looking like how it looks in this picture. And finally, the last matrix that we collected was any seaweed, seagrass, or macroalgae along the rack line at the beach during that sampling period. And interestingly enough, we planned to finish this study right in March of 2020, but given that our last sampling period coincided with the week that everything changed and the world shut down, we were given the opportunity of sampling the beaches where no people or no human people were allowed to go to the beach. So you really saw the drastic difference in terms of before you have people at the beach, operation as normal, people bathing, and people can go to the beach and enjoy their day. But then you also have this drastic change of no people at the beach. All you see are tire tracks along the shore, and that's just from any ATVs that the beach managers were operating just to make sure that people stayed away from the beach. And then after, we have that influx and that pulse of people coming to the beach, and just having that increase, seeing if there's an increase in bacteria overall. So within their time periods of before, during, and after, we saw that we collected samples over 27 sampling periods, and 13 corresponded to before, 6 corresponded to during, and 8 corresponded to the after time periods. So we really wanted to see if humans impacted the beach environment in such a drastic way, specifically at Key Biscayne Beach. So for water and sediment, we did collect 39 samples before, 18 samples during, and 24 samples after the beach shutdown period, respectively. And we were fortunate to see influxes of seaweed strandings. And these pictures don't do justice just to the amount of seaweed that does make its way onto shore. So we did have one sampling period correspond to a seagrass bed die-off, and this was during the shutdown. And this is only just a snippet of just the extent of seagrass that made its way onto shore. We also had 2 seaweed stranding events that corresponded to after the shutdown, and June 23rd, you could just I remember when I sampled, the seaweed just kept rolling in and it had nowhere else to go. And at the time, what they do is they just take a beach raker and they go back and forth across the sand with the seaweed, and the sand just can't take any more seaweed. You can see just the clumps of seaweed in the sand. July 21st is not as drastic as June 23rd, but again, you still see just these large amounts of sargassum making its way onto shore. So this also just provided us an opportunity to really see what is happening specifically in the beach environment when you have these large influxes of seaweed or seagrass on the shore, and how it can interact with the sediment. So after those large seaweed stranding events, we did sample a week later, but you could still see the remnants of the seagrass or the seaweed or sargassum that's on the shore. So yes, Key Biscayne does try to integrate it, and they only integrate in the morning hours from about 6 a.m. to about 10 a.m. So throughout the day, you may have more seaweed just making its way onto shore, and they are not taking care of it because they only take care of it within the morning hours. So you can see this amount of seaweed that came in after they maintained the beach, and you can see just the decomposing of the seaweed within the sand, especially on June 30th. To kind of go into the results from the study, for our water samples, specifically for Enterococci by culture, we did see that there were no statistical differences observed in the water before, during, and after the shutdown. But we did have elevated levels observed during the shutdown, which were driven specifically in the knee depth water on one of the seaweed stranding events. So this level, again, is characterized just by that outlier that we had during our seaweed stranding event. Just to kind of further evaluate the data, I did mention that Miami-Dade Department of Health has been monitoring this beach since 2000, so we had access to this extensive data set to kind of see what is going on with the bacteria levels and how they kind of compare with the data that we collected. So we broke down Miami-Dade Department of Health data into weekly data, which matched up with our data set. And then we also looked at their weekly data, which just took place since I looked at it from a year prior, from about 2018 to current. So that's what is presented to you here. So you can see that in all cases, the lowest levels were during the shutdown in the waist depth water for all three sets of the data, and intermediate after the shutdown, and then highest before the shutdown. To look at the culturable data for Enterococci within the sediment zones, again, I broke it up into the supertidal, integrated, intertidal, and subtidal. So overall, the results show that bacteria levels decreased during the shutdown, except for the integrated sand, and then it increased after the shutdown. So these differences were particularly apparent within the supertidal zone, where the levels before the shutdown were higher than both during and after the shutdown. So like the supertidal zone, the intertidal zone did experience similar trends, and then levels were higher within the zone for samples collected before, during, and after the shutdown. So over the course of sampling, there is a guideline or threshold that is outlined by WHO. So the World Health Organization says that anything that's over 60 CFU is considered high for sediment samples collected. So we did experience six of those exceedances. So 5 of them were observed in the integrated sample. So that goes to show you that when you integrate your sediment and your seaweed, you're going to get higher levels of bacteria, as evident from the 5 exceedances above the 60 CFU per gram that is outlined by the World Health Organization. So within our seaweed samples that were collected, we broke it. When initially we collected these samples, we noticed that there were differences in moisture content. So given that there were differences in the moisture content, we decided to break down the seaweed samples into what we dubbed a freshness scale. So we considered fresh seaweed anything that has a moisture content of 74% or greater. Senescent seaweed ranged from about 55% to 74%, and decomposing seaweed ranged from anything below 54%. So for Enterococci by culture, for all three of the freshness categories identified, concentrations were lower on average during the shutdown period compared to before and after the shutdown period. And then differences were observed during and after the shutdown for the decomposing seaweed. For fresh seaweed, bacteria levels were also statistically different during and after the shutdown. Senescent seaweed did not exhibit any statistical differences before, during, or after the shutdown. Within this study, we also looked at chemical source tracking, but we only used chemical source tracking on any of the samples that had an exceedance of the 70 CFU per 100 mil or greater in the waist depth water, because again, waist deep water is where the Miami-Dade Department of Health also collects their samples. So we did experience 3 exceedances of the 70 CFU. We had 1 before the shutdown and 2 after the shutdown. And we looked at sucralose, naproxen, ibuprofen, acetaminophen, and caffeine. And these samples were collected and shipped out to an outside laboratory for analysis. So from our chemical source tracking results, we can see that caffeine is consistent in all 3 of the samples that did exceed the 70 CFU per 100 mil threshold. So you can see that there on March 9th, which was before the shutdown, we have about 14 nanograms per liter. And then you can see that after the shutdown, you do see an increase as well. So since we had caffeine being consistent in all 3 of the samples, we believe that there is human sources specifically on these days. We did also detect sucralose in one of the sampling periods after the shutdown, and we also detected ibuprofen in one of the sampling periods, again, after the shutdown. So from our, again, part of the study, we looked at microbial source tracking because we want to specifically pinpoint down where is the source of bacteria coming from, and is there anything that can be done to minimize this source of bacteria to the beach. So as mentioned, we did do culturable Enterococci, which is outlined in the EPA method 1600, but we also looked at qPCR and looking at just general Enterococci. So we used the assay Entero 1A, which is outlined in the EPA method 1611. But specifically to the beach environment, we did look at 2 human markers, HF183 as well as HUM-M2. And we also looked at seabirds, which is the assay used is Gull2, and we looked at dog as being a source of Enterococci, and in which we used the assay dog back. So even though the village of Key Biscayne does not allow dogs at the beach, the picture that I showed you previously with dogs at a beach was taken at Key Biscayne. So we wanted to see whether or not the amount of dog bacteria can be found and detected in our beach samples collected. So within our samples collected for MST, for the Entero 1A, which is just a general Enterococci based on qPCR, you can see that the levels were lowest during the shutdown compared to before and after the shutdown in all 3 matrices. For sediment and seaweed, we did see that there were statistical differences. So again, any of the samples that are sharing a subscript means that they were not statistically different from the other time periods. For the human marker HF183, we did have low levels observed during and after the shutdown compared to before the shutdown in all 3 of the matrices. For the human marker HUM-M2, we did see that there were low levels or either below detection or what we called our environmental lower limit of quantification during the shutdown compared to before and after the shutdown. For the seabird or the Gull2 marker, we had the highest levels during the shutdown compared to before and after the shutdown for the water matrix specifically. And then for the sediment, we had higher levels noted before the shutdown. And finally, for the dog marker, we saw that the highest levels overall were observed before the shutdown compared to during and after. And most of our samples were either below detection or not detected at all for the water and sediment sample. But for the seaweed matrix, you do see that there's an increase after the shutdown in terms of the amount of bacteria that was detected using qPCR. So what can we conclude from this first study? Specifically for our water samples collected, we did see that cultural bacteria levels in the water were higher in the knee sample during the shutdown compared to before and after the shutdown. And during the shutdown, there were large influxes, as you can see from the pictures I showed previously, of seaweed on the beach and in the water. So the picture that you see on this slide was taken after a large seaweed influx, and you could just see the turbidity and the bits of seaweed in the water. And this does also contribute to the levels of bacteria that can be detected within the water sample. So when there is an influx of seaweed, you can't get away from the bits floating around. So this does impact the bacteria levels that it's at the beach. So within the HF183 marker, it did exhibit the most statistical differences between time periods and water depth across the sampling locations. So averages before the shutdown were higher compared to during and after the shutdown. And this can just be attributed to the number of bathers and humans and people that make its way to the beach. Within the sediment samples, what we concluded is that human presence on the beach was definitely a dominating factor, and it's associated with higher enterococci levels in the sediment outside of the integrated sand zone. And again, that integrated zone is when you combine your sediment and seaweed and you have that stark difference within the beach environment. So this was evident from the cultural bacteria levels observed in the various sediment zones, especially for the supertidal sediment across time. So the fecal indicator bacteria is believed to accumulate and proliferate within the supertidal zone, since it does have optimal conditions such as the temperature and the moisture content for preferential growth. So whenever we collected the temperature of the supertidal zone, it did tend to match up with the temperature that is used for incubating the bacteria within the lab. I believe we had a temperature of about 40.4 degrees Celsius within the supertidal zone, and the temperature that we use for incubation is about 41 and give or take 0.5. So it is relatively close to the temperature that is used to incubate bacteria. So the supertidal zone just provides that temperature that is used to incubate the bacteria in the environment. We did also see that there were lower levels found in the intertidal zone and the subtidal zone, and this can just be attributed to just the constant wave action where these zones are located. Because again, the intertidal zone is where the shore meets the water, and you just have that constant wave action and flushing within this zone. And again, you just have the bacteria is not allowed to, like, let's say, stay and proliferate in these two environments. Interestingly, for our seaweed, because again, there has been very limited research conducted on seaweed overall as being a source of bacteria to the beach. And just to give you an idea of what a fresh seaweed sample would look like versus a senescent and decomposing, you can see the fresh is that nice golden tide that that comes onto the shore, so it's nice and fresh, and it has very high moisture content. Within the senescent seaweed samples, it's starting to decompose, but it's not quite dry and crispy, as you can see what a decomposing sample would look like. You can see just how it looks very brittle, so you're able to just crush it with your hands. So you can see just the difference between the fresh, the senescent, and the decomposing seaweed. So as the seaweed decomposes on the beach, it does also act as an insulator, and the temperature begins to increase overall. So as similar that we saw with the sediment in the supertidal zone, the temperature is just that optimal temperature that allows the bacteria to proliferate. We're seeing something very similar with the seaweed samples within the seaweed raft that we collected. So with the increase in temperature, the bacteria is allowed to proliferate as well. And in addition to the increasing temperature, the seaweed itself can act as a substrate for the bacteria to grow. So this just gives more fuel to the fire in terms of the bacteria proliferating and bacteria constantly being found in the environment. We did see that there were high levels of the Gold 2 marker, or the seabird marker, within the seaweed samples. And this can just be attributed to the mixing of the seaweed and the sand, and just how birds, anything that can get trapped in the seaweed as it makes its way onto shore, any insects or anything that may die and birds may be attracted to. So this is why we think we have higher levels of the gull marker or seabird marker within this seaweed zone. So any of the organisms that live in the seaweed can serve as a food source, which will then attract the birds, which will then give you higher levels of the Gold-2 marker. And it just, again, the seaweed provides that nice environment for the bacteria to proliferate and regrow within the environment. So given the results that we saw with our seaweed work within the Village of Key Biscayne project, we identified this as being a source of enterococci to that beach. So what can we do when we have those large influxes of sargassum? Is there anything a municipality can do to try to help relieve the amount of sargassum that makes its way onto the beach? So we did start to evaluate composting as a way to recycle the sargassum on the beach. And this project was funded by the Hinckley Center for Solid and Hazardous Waste Management and was done in collaboration with the City of Hollandale Beach. And these pictures were taken at the City of Hollandale Beach. So when we have an influx of sargassum on the beach, we plan to collect it and then to create a compost pile and see what the qualities of compost are. So with that in mind, we set up two different experiments. We looked at small scale as well as large scale experiments. So within our small scale experiments, we looked at tumbler composters, which specifically just looked at evaluating additives to sargassum, specifically grass and mulch. And within our large scale compost piles we set up with the City of Hollandale Beach, we just looked at adding vegetative waste and no additives at all. So some of the compost parameters that we looked at and we monitored, again, we just looked at both physical and chemical parameters. We looked at nutrients, we looked at metals or elemental concentration, and we also looked at bacteria analysis, specifically enterococci and fecal coliform, because from our study within the Key Biscayne study, we identified that there were high levels of the bacteria within the samples. So with our small tumbler composters, we again just looked at 4 different recipes. We looked at washing the sargassum, unwashed sargassum, so just taking the sargassum and adding it to the tumbler composter and leaving it as is. We looked at adding sargassum and grass clippings, as well as mulch and sargassum. And these tumbler composters were sampled biweekly for those bulk physical and chemical parameters, the bacteria, the elemental concentration, as well as the nutrients. And I just want to point out, you can see the top row of pictures is what the tumbler composters initially looked like at the start of the study. And then you could just, again, see that drastic change in just the sheer amount of sargassum that we're left with. So there's about a 90% reduction, as you can see from the top row of pictures versus the bottom row, which is the end of the composting. So when we looked at the bulk physical and chemical parameters, we specifically just looked at the temperature, the moisture content, pH, and the conductivity. And we broke it down into beginning, middle, and end time periods. And these red boxes and lines represent the U.S. composting guidelines and what they outline as being ideal compost. So within our moisture content, we are not within what is recommended by the U.S. Composting Council for the moisture content, as well as the pH and the conductivity. So when we initially started this project, we did think that the conductivity was going to be a issue, because again, you're taking something from the saltwater environment and you're composting it. So we did believe that the conductivity was going to be an issue, especially when making compost from sargassum. And again, from our Key Biscayne study, we wanted to just see how the levels of enterococci and fecal coliform can change over time during our composting process. And that red box that you see within the fecal coliform graph is the limit that's outlined by the U.S. Composting Council. So they recommend anything less than 1,000 mpn per 100. So you can see that we did not reach the recommended guideline by the U.S. Composting Council within the different piles and recipes that we created. So we did as we predicted from the cubic gain study, have higher levels of bacteria within our compost sample. For the carbon and nitrogen ratio, again, this red box is what's outlined and recommended by the U.S. Composting Council. Our recipes did not meet those guidelines as well. And going on to the metals and elemental concentration analysis, we did this in 2 different ways. We used an XRF or X-ray fluorescence on our compost samples just to see what elements we were able to detect. And we did see that strontium and iron were consistent in the 4 different recipes that we tested. And we also noticed that zinc was only noticeable in our grass clipping sample. And interestingly enough, we were able to detect arsenic in our unwashed and mulched samples. So given that we were able to detect arsenic and just kind of going back and forth with our technical awareness group, we did identify that this is a concern, and a lot of the operators and municipalities are going to be interested in looking at just the arsenic concentrations in Sargassum and how does that change over your composting process, and is it going to be that big of an issue, given that we still do detect it. Just to kind of give it into the finer details of the elemental concentration, we did also utilize ICP-MS. So the compost samples were digested using hydrogen peroxide as well as nitric acid in collaboration with Dr. Amanda Ehlert out at Rasmus. So we did digest our samples and we did look at the elemental concentrations within the compost samples collected. There's a lot of information on this graph, but just to point out again, arsenic was something that our technical awareness group was really interested in, and they wanted to see how what our ending concentration is. So over the time periods of beginning, middle, and end, in all 4 of our recipes of washed, unwashed, grass clippings, and mulch, we did detect arsenic. So in a graph later on, I'll show you how these levels of arsenic does compare to what's outlined by 4 different regulatory agencies. And just to see if we create this usable compost, can it support plant growth? So in order to test that, we created 5 different experiments, and we have these radish bioassays that we use to test this theory. So we looked at 5 different experiments. We have just 100% potting soil. We have 100% compost. We have a 50-50 mix of compost and potting soil. We have a 70-30 mix of compost and potting soil. And we have a 30-70 mix of compost and potting soil. And again, from these pictures, the far left represents all of the compost that had the mulch additive. Next, we have the grass additive, we have unwashed, and then we have washed. So you can see the progression over time. We did see plant growth, and the best one that had, I guess, the best compost mix that had the best plant growth was our grass and Sargassum additive. So given that we did have better plant growth within our grass and Sargassum additives, we decided when we were setting up our large compost piles with the City of Holland and Beach, we did just want to look at pure Sargassum versus Sargassum and vegetative waste. And again, from the pictures, you can just see the drastic decrease in the amount of Sargassum that you start off with versus what you ended off with in terms of our end product. And again, just looking at the physical and chemical parameters, we do see that very similar to what we saw with our tumbler composters, we weren't within the recommended range, what outlined by the U.S. Composting Council for the moisture content, the pH, or the conductivity. But interestingly, for the conductivity, we did start off at a higher conductivity, but then we did see a decrease overall. And we suggest that, again, when these large compost piles are created, they're exposed to the environment and they're just exposed to the open. And we did have a lot of rain, so that can just allow the natural washing of the Sargassum. So we did see that there are lower levels of the conductivity within the sample. And very interestingly and surprisingly, for our fecal coliform, again, that red line at the bottom is what's recommended by the U.S. Composting Council, and anything less than 1,000 MPN is what they recommend. We did achieve this within our large-scale compost piles. Enterococci is not regulated or is not looked at by the U.S. Composting Council. They look at fecal coliform, and we did reach what is outlined by the U.S. Composting Council. For our carbon to nitrogen ratios, again, we did not quite reach what is recommended by the U.S. Composting Council. And with our XRF analysis in our large compost piles, we were able to detect more elements with this analysis. And again, we do see that strontium and iron, again, are consistent in both of the piles. And we did detect arsenic in the sargassum only pile. So when the sargassum was mixed with vegetative waste, we did not detect arsenic. And again, this graph is also a lot. This table is also a lot. But just to make it very simple, anything for the small scale tumbler composters that is outlined in that light pink shade does reach what is recommended by the U.S. Composting Council, as well as the U.S. EPA guidelines for biosolids. So by these guidelines, we are within the arsenic concentrations that they outline. Within the five other metals, you can see that we are within what is outlined by the U.S. Composting Council, the U.S. EPA, and the FDEP criteria for compost, as well as the soil cleanup target levels. And I also just want to point out, we were not within the bacteria levels for the small scale tumbler composters, but we were within the bacteria levels outlined by the U.S. Composting Council for the large scale piles that were created. So what did we learn from this study? So we learned that there was almost a 90% reduction. Again, you can just see that drastic difference in what our start product was versus our end product. And we initially thought that salinity was an issue, but we did not detect that salinity was an issue, especially given that we were allowed to grow plants within the compost that was created with the tumbler composter. Again, arsenic did meet the U.S. composting guidelines, as well as the U.S. EPA guidelines for biosolids. So arsenic did meet 2 of those 3 standards, because arsenic is not regulated by the FDEP for criteria for compost as of yet. We did see higher levels of bacteria in the tumbler composters, and we had lower levels of bacteria within our sargassum large compost piles created with the city of Hollandale. And again, these piles were open to the environment, so you had that solar inactivation, whereas within your tumbler composters, it's closed and it's dark, and you don't really have much of that solar inactivation being present. So overall, it is a promising solution, and we were able to use the compost to grow radish seeds, and we'll see what elemental concentrations are within our radish plants. With that, I'd like to thank our collaborators, Dr. Amanda Ayler and Dr. Peter Swart, for our composting project, as well as the City of Hollandale Beach, Alyssa Jones-Wood. And for our Key Biscayne project, I'd like to thank Dr. Chris Senegaliano and the Village of Key Biscayne for funding the project. And with that, I'll take any questions.
Thank you very much for an excellent presentation, Afeefa. Let me open it up for questions from the audience. If you have a question, just go ahead and raise your hand and I'll call on you. While everybody's thinking, let me just ask a question then. What statistical tests did you use to determine the significance of the difference?
Within the Key Biscayne work? So
Yeah.
within the Key Biscayne work, we used 2 different